All right, so then when we move on and we have to figure out what the most probable genotype, I keep saying that, sorry, what the most probable genotype is going to be based on reactions, I'm going to give you guys this sheet. You have it already. This is the prediction of the Rh genotype. And on the back, it has all of the frequency, so the probability of the North American population, like white Caucasian North Americans. This is what we're most frequently seeing. Remember that depending on your geographical location, the probabilities can change. There's this little asterisk that we see some of these more commonly in Black people than we do with white people. So it can, depending on the part of the world that you're working in or the type of patient population that we're getting, you could see different frequencies for sure. But this is just the setup for Caucasians. Okay, so there's a couple of ways we can tackle this, and I will ask this question in several different formats. So like I said, I do want to kind of get into the antigen testing first and then show you how we can predict. So this is something that we actually do in the hospital. So like I said, you can have the anti-sera and test the patient's red cells with all of these different anti-seras. This is just like the forward grouping, guys. We just covered the forward grouping in great detail. It's the same idea. You have a reagent known antibody, the anti-sera. You're mixing it with patient red cells, and then you're going to see what antigens are present on the red cell surface. And it tells you if you're positive for that antigen or negative for that antigen, just like the forward grouping. So instead of calling this anti-A and anti-B, we're going to have anti-D, anti-C. I'll do it in the right order this time. Anti-big E, anti-little c, and anti-little e. So when we test for these, we actually set up the tubes and we're going to grade the reactions just like normal. So let's say we have a positive reaction in all of them. So we have a nice strong 4+ reaction in all of these. That means they have every single one of the Rh antigens. So when you take your sheet about predicting, you guys are going to have to learn and practice this on predicting and just following the rules. If you follow these rules, this is how we're going to predict the most probable genotype setup on which chromosome has which antigens or which chromosome has which genes. And that way we can predict what the offspring could be that way. So when you follow the rules, the first thing it's going to say is the big D is always on the first chromosome. So this is like what we do in the lab. Like I said, we phenotype the patient. We have all of their antigens, and we see which ones they have. And in this example, they're going to have every single one of them. So then we're going to set it up, and this is how I've always done it. I always like to take 1, 2, 3 chromosomes here, or sorry, 3 spots on the first chromosome and 3 spots on the second chromosome. And then you follow along with the rules. So the first rule, if you follow that sheet, says most probable Rh positive for Rh positive patients. So we know they're Rh positive because they have the D antigen, right? We use anti-D and it came up positive. So this is the most probable for an Rh positive individual. So big D is always on the first chromosome. So when we set it up like this is chromosome 1, this is chromosome 2. So D is always on the first chromosome. And I always put it first. I don't put them alphabetical. I always put the D first because it's the most important one that we're looking at. So the D goes on the first chromosome. So we put it here. And then the second step is going to say look for big letters, like look for capital letters. If patient has both the big C and the big E, then the big C goes on the first chromosome. So we have big C and we have big E. So big C goes on the first chromosome and big E goes on the second. And now we're going to put them in their correct order, their correct position. So I always do DCE, DCE. And that's the order we'll put them in here. And then it says E goes on the second chromosome. Never put both big C and big E on the same chromosome for the most probable. So yes, we can have somebody who is DCE on one chromosome and DCE on the other chromosome. This is a possibility. I'm not saying it's impossible. As long as you have the letter D, the letter C, and the letter E on one chromosome and then another D, C, E on the other, you can't have D, C, C, right? We already talked about that. You have to have one for each. Honey, can you not do that for a sec? So as long as you have one letter for each spot, then you're okay. So yes, it's possible. You are able genetically to have D, big D, big D, big C, all in the same big D, big C, big E, all in one chromosome. That is genetically possible, but not the most probable. And that's what the order we're working in. So we're going for the most probable. So if you have the big C and you have the big E, you're going to separate them. And that's the only way you know if you have the big C or the big E, if your patient has the big C or big E, is you have to go back to your antigen testing. Look to see where they came up positive. If they have the antigen, then we're going to fill in their genetic makeup like this. Okay, so we covered the capital letters, and then it's going to say, this part's actually pretty easy. Once you get this far, it gets pretty easy after that because you have to now fill in your other spots. So regardless of what we can see over here, we know that this is the E spot, and we know that this is the C spot. So you have to be able to fill in the C and fill in the E, fill in the E here and fill in the C here. So it's just a matter of it's going to be a capital letter or a lowercase letter. So we go back and we look at what's left over. We've already used up the big C. We've already used up the big E. They're both present and we filled them in. So now we know that there's a little c and a little e. So the only spot left for those to go, the little e has to go here for it to be present, and the little c has to go there if it's present. So just by process of elimination, you know that that's where they're going to go. But if you look at the rules, if you need to follow the rules properly, it'll actually tell you, look at the little letters. If the patient has both little c and both little e, the little e goes on the first chromosome and the little c goes on the second, because you're putting the opposite with what was already placed. You have to fill in the blanks. So to me, you don't have to follow the rules. You can just fill in the blanks, right? If you know the patient has those other two antigens, they have to go there. There's no other spot that they can go. But if you need to follow the rules, go look at the rules, and it will tell you that if you have both little e and little c, the little e goes on the first chromosome and the little c goes on the second. And then finally, it's going to tell you that, it'll say if the patient only has the little e, then obviously the little e is going to have to go on both chromosomes. So if the patient only has little e and not the big E, so say that this came up negative, if they didn't have it, obviously this would have to be a little e over here. And if they didn't have the big C, obviously this would have to be a little c here and a little c here. So some of them are easier than others to fill in because you just don't have anything else to use. Like there's just nothing else to put there. If they don't, and I'll show you another example of that later, but it's those are the easy ones. When you have every single Rh antigen, that's when it's difficult to predict. But when you're lacking some of the antigens, meaning like if they were E negative, big E negative, then the only thing that can go in those two spots are the little e, right? And remember, for inheritance on a chromosome, you have to in order to be a proper Rh person, you have to have all three spots filled in. The D, the C, and the E. The D, the C, and the E. So you're going to have either the big D or the little d. Remember, the little d is kind of like a silent, it doesn't exist. But you'll have something there. And then for the C, you're either going to have the big C or the little c. You can't have nothing. If you have nothing, you're an anomaly. We are, of course, there's always exceptions to rules. So we will talk about the Rh null people who actually don't have anything there. But for normal, everyday people, we have to have a spot for the C, and it's either going to be a capital C or a lowercase c. And you'll either have the big E or the little e. So you're going to have positive reactions somewhere up in these Rh. You're either going to have all of them, or you have to have at least the little e and the big C, or at least the little c and the big E, or at least the little e and the little c. Like you need to have at least one of the above, or I guess two of the above. You need to have one of the E portions and one of the C portions. So do not fear, they will come up positive somewhere. And then finally, so we talked about whether or not you have your C's or your E's, whether they're capital or lowercase. And then it says, if you have a big letter on the second chromosome, this is our second chromosome, number 2. If you have a capital letter listed here, which we do, we have a big E. It could be a big C or it could be a big E, but if there's a capital letter present on that second chromosome, then the D portion of it is most favorably and most probably going to be a capital D. Capital D, like the D antigen prefers to pair up with another capital. Now, yes, it is possible, like genetically, you can be capital D, lowercase c, lowercase e. Of course, that is a possibility. Somebody could have this genotype, but the most probable is that the D, or sorry, if there's a capital letter on either one of those, so even if we're capital C, little e, doesn't matter, there's a capital letter there, the D is most favorably going to hang out with another capital letter. So if there's a capital there, it's probably a capital there too under the D. That's for the most favorable and the most probable. So that's our most probable genotype if this is what the patient's reactions were. So if the patient came up positive with every single one of the RH antigens, then genetically, this is probably what their chromosomes look like. Okay? So the other option, like if we didn't play the rules right, we could just randomly pick and say, well, they have the D, they have the big C, and they have the big E, and they have, well, again, we said they have the big D, and they have the little c, and they have the little e. So we could have written it like that, but the probability of 3 capital letters hanging out on one chromosome and two lowercase here with a capital D, the probability of this is very low. There's usually not 3 capitals statistically on the same chromosome. They usually spread them apart. And when you have 2 lowercase letters on one chromosome, 2 lowercase c's and e's, then most probably the D likes to hang out as a lowercase as well. So like I said, we could have predicted it. Sorry, let me put it back the other way. We could have predicted it just by going down the line and saying, hey, maybe this is their genotype. But that doesn't match up with what we wrote. We swapped the E's. Oh, that's not, I wasn't supposed to put it, that's supposed to be a D. We swapped the E's. And we swapped the E's. We didn't fill it in the most probable way. So don't just guess and fill in the blanks. You have to follow the rules that I gave you guys and fill in the blanks as you go through and follow the rules to get your answer. So you could actually do this and we will next year in lab. I actually make you guys test this. We test every single one of the RH antigens. You get your results, and then I make you predict what their most likely genotype is going to be. So if we did this again for another example, and this time I'll remove one of the, I'll make one of them negative. So if we do it again, and we actually take our tubes and we have anti-D, anti-C, anti-E, anti-little c, and anti-little e. And our reactions are, let's say a 4 plus, 4 plus, 0, 4 plus, 4 plus. Then we have to predict. So I do my same thing like I always do. I go 1, 2, 3 on one chromosome, 1, 2, 3 on the other. That's how I start. And then I say, okay, I have to fill in the blanks. So the first question is always, if they're Rh positive, if the D is present, where does the D go? The D goes on the first chromosome. So we have the D present. So the D goes on the first chromosome. And then it's going to tell us to look for capital letters. Do we have a capital C? Yes, we do. Where does it tell us that the C usually goes? The C likes to hang out on the first chromosome. And then it's going to tell us to look for the lowercase letters. Oh, I put the C in the wrong spot there. Sorry, guys. The C, it's still on this first chromosome, but I like to put it right here. And then look at the lowercase letters. So we do have the little c and the little e. So at this stage, because we're missing the big, it would have told us, like, if there was a big E, look for capital letters, the big C goes on the first chromosome and the big E goes on the other chromosome. But we don't have a big E, so we can't put a big E here. The only E we have is a lowercase. So this one's easy. We're literally just going to put a little e there. And the other little e has to go here too, because there's no big E at all, no big E to fill in. So both spots have to be filled up by this nice positive right here, little e. So we'll fill those in. And then the only thing left that we haven't filled in yet is the C on this side. So if we were to just put our big C here, well, we have no spots left for our little c. But our little c came up positive, so we need to fill it in. So this is going to tell us this is the little c spot. And then if you read through, if you read through, it's going to tell you, I'm trying to find the right spot, where it is on the thing, but it's going to tell you that if you have if these are two lowercase letters. So remember when I said earlier that you look on the second chromosome, this is chromosome 2. If there's a capital letter here, then this D is going to be positive, a big D. It's going to be a capital D. But the alternative to that, we don't have a capital letter here, do we? We have two lower cases. So it told us if there's a capital letter here, then this is going to be a capital D. But since there's not a capital, then the most favorable, like the most likely, is that this is going to be a lowercase d. Because like I said, the big D likes to hang out with other capital letters. And if there's not one there, then the most probable genotype is that this is going to be a lowercase. Okay? And that's how you fill it in. So it is actually easier when you're missing a result. And we can do it one more time. We can fill it in one more time with a different result, like if I made this a negative here. So let's follow the rules again. It's going to say if there's a D, a capital D, put it on the first chromosome. We have a capital D, a big D, so we'll put it on the first chromosome. Then it says look for capital letters. Do we have any capitals? Do we have a positive big C or a positive big E? And we don't in this example. So this one's really, really easy. So we put the D on the first chromosome. That's correct. We have chromosome 1 and chromosome 2. Put the big D on the first chromosome. That's correct because there's a D present. But now we don't have any capital letters. But we still need to fill in the C and the E sections. So what do we fill them in with? We only have the little c and the little e for positives. So we'll put a little c and a little e and a little c and a little e because there are no capitals at all to fill in on either chromosome. And then what was the rule that I told you just before this? You look for a capital letter on the second chromosome. If there is a capital letter on the second chromosome, then that D will be a capital D. If there is no capital D, then it's most likely going to be a lowercase, or sorry, if there was no capital letter, no capital C or capital E, then it's most likely going to be in this expression. All the lowercase letters are going to hang out together on one chromosome. So here we have an example of the capital D hanging out with a lowercase c and a lowercase e. Same thing, I just rewrote it. But it's because the D is present. So this is a very rare phenotype that we're looking at. The fact, like phenotypically, the fact that they have this pattern of antigens on the red cell surface is not common in North America. So for white Caucasians, this isn't normal. This isn't common. But if you went to other parts of the world, it would be more common in their genetics, but just not so much in North America. But it's still genetically possible. I'm not discounting it as a possibility. So here's an example of where you will have the D as a capital with 2 lower cases because you followed the rules. The first rule says if the D is present, put it on the first chromosome. And we did. But then you don't look at the second chromosome until you filled in the other letters. We looked for capitals and we didn't have any capitals, so we had to fill in the C and the E spots with whatever was left over. And what's left over are the 2 lowercase. So lowercase, lowercase, and then we did lowercase, lowercase. We didn't have this filled in yet. And then we said, okay, what's the rule? If there's a capital letter here, then this is most likely a capital D, but there is no capital, so the most likely result is that that's going to be a lowercase d. And that's how you predict. So you guys can do this yourself over and over and help, you can help predict what the most likely result's going to be. And again, it's not a guarantee, but it is how we like to predict it with the patients that we're presented with. And it's not foolproof, but it's a good estimate when we need to try and estimate sometimes with our fetuses that are potentially at risk for suffering from hemolytic disease in the newborn. So if we're not properly lining up our genes, our 2 chromosomes, then we might be screwing up our results with our probability for our patients. But it's not a guarantee, so it's not an end-all be-all if we get it wrong, because we could have just gotten it wrong. But if you look at this sheet on the back, so you have your most probable predictions, and flip it over, we have all these results. It says reaction with anti-sera. That's what I'm doing here. Anti-sera is anti-D, C, E, little c, little e. And that's exactly what I did here. So we have positive reactions and negative reactions, just like what's listed there. Then it's going to turn it into the Fisher race and the Wiener and the frequency of that type of phenotype. And again, what we were just doing where we had D, C, E here, and then we had D, C, E here. That was the last one we just did with that. You guys can then, for practice, turn this in to the Wiener notation. So this would be R with a 0 because it's a capital D, but no uppercase. So R 0 and little r. This one's my favorite. This one's so easy to remember. If there's no capitals whatsoever, it's just little r. So we went from antigen testing to Fisher race to Wiener. And we can put them all together. And that's why it's important to understand all 3 nomenclatures so that you can make sense of them all and help them with your predictions. So I could ask you on a test to go convert from this to this to this, or to go from this to this and tell me what the likelihood of that offspring having this antigen is. Or I could ask you from the Wiener notation, what's the probable antibody that this patient could develop? So if this is what I give you and I say, what antibody in the Rh blood group system are they most likely to develop? Do you know how to figure that out? You'd have to convert this back to Fisher race, which we already have up here. And then you have to say, if I'm asking you what is the most likely Rh antibody that this patient could develop, how do you develop an antibody? You have to be lacking the antigen. So what Rh antigen are they lacking here? Well, sometimes this is hard to see, so convert it back to Fisher race. If you convert this will be on the test question, but you have to be able to, in your head or on paper, convert this back to Fisher race and say, which Rh antigen are they lacking? Well, they're lacking the big C, not with this. Don't use this as your clue because you won't have this on the test. They're lacking the big C and they're lacking the big E. So this patient would be capable of developing in their plasma an anti-C and an anti-E. They couldn't develop a little e because they have the little e. You don't develop an antibody to an antigen that you already have. But they're lacking the big C and they're lacking the big E. So they could develop an anti-big C and an anti-big E. All right, so I think that's all I really need to do for this. I could keep doing them over and over again, but you guys just need to be able to practice. So even without looking at your sheet, you can just take this information, write out anti-D, C, E, little c, little e, and just make up results. There is another way when we're looking at it, if they are... if they don't have the D antigen, like if they're lacking the D, you'll still just kind of follow along and put your antigens in the right spots. So like, for example, it tells you along here when you're reading through your sheet, if you have to change your prediction, the second most probable after we just worked through what we worked through. We went through number 4 and it says if you have a big letter, a capital letter on the second chromosome, as long as there's a capital letter there, then you're most likely going to put in the D slot a capital D. And if you don't have a big letter, then the D portion is most likely to be little d. And that's what we had just said. If we filled this in, we had little c and little e here, then the D is most likely to be lowercase. That filled in number 4. And then the second most probable is switch the D, the big D, to a lowercase d, or vice versa on the second chromosome. So if you had to update your probability, like change your probability, you would be updating the second chromosome first and switching the Ds. Third most probable is switch the big letters from the first to the second. That's supposed to say from the first chromosome to the second chromosome. So we can swap out the big letters and always have the little d on the second chromosome. Fourth most probable, this just keeps going on and on. And it just tells you how to manipulate your genetics. We don't really work that far down. What I always ask you guys is what's the most probable? That's what I'm always going to ask you. I don't care what the third most probable is. We always base on probability and what's the most likely. So you can fill those in. So just keep practicing with this, and depending on what's on your antigen testing sheet, like when you actually set this up and you do your antigen testing in your tubes, based on whatever results you have, you can change these and you guys can just all practice and swap everything out. And you can put in 4 plus 4 plus 4 plus 0, 0. The only thing is you have to make sure that at least the C and at least the E portions are filled in some way, whether it's a capital or a lowercase. D always goes here, look for 2 capitals, and we have 2 capitals. So you put them in on C on the first, E on the second. Then we don't have any lowercase, so this has to be a capital, and this has to be a capital. We have no other options. And then if you have capital letters here, then this is most likely a capital D, and we have R, Z as our Fisher, or as our Wiener notation. So just keep filling them in, keep changing these up, keep practicing and filling in what you think the possibilities are. And you can then convert, for practice, convert what you have from Fisher race. Without looking at your sheet, try and convert what you have from Fisher race into your Wiener notation. And that will make sure that that'll help you guys practice the conversion as well. So you go from one to the next to the next.